Welcome back, everyone. Next, we have CytoMed Therapeutics. They trade on the NASDAQ under the symbol GDTC. It is a biopharmaceutical company that focuses on harnessing its licensed proprietary technology to create novel cell-based immunotherapies for the treatment of human cancers. Please welcome its chairman, Peter Choo, and co-CEO and COO, Dr. Tan. Welcome to the conference, gentlemen.
Hello.
Hi. Thank you for having us.
All right, the floor is yours. Call me back when you're ready for questions.
Thank you, Anna. A very good morning to our American shareholders, partners, ladies and gentlemen, and a very good evening to those in Asia. We are based in Singapore, and we are actually in our Singapore lab now. We are 12 hours ahead of Eastern Standard Time. So my name is Peter Choo, and I'm the chairman and the major shareholder. And with me is Dr. Tan, our co-CEO and COO. Let me now jump straight into the presentation. If you have got interesting ideas, I will be in New York in the last week of October to meet anybody who would like to have a face-to-face meeting. If you have any question, you can email us separately and we can converse. Next is the standard disclosure as a disclaimer. Well, so CytoMed, what are we? We are not a diagnostic company. Right? We don't make diagnostic kits to test cancer and the presence of diseases. We are also not a research biotech, right, focusing on target discoveries, which burns cash. So we are not that. We're not target discovery. We are also not a research biotech focusing on contract development, manufacturing. They call it the CDMO or CRO, providing contract manufacturing development. So we are also not a medical device company. What we are is we are focused on cell therapies. Right, working with our partner healthcare providers in Southeast Asia. So our mission is as stated in this slide. We engineer, we make cell therapies and make it affordable as well as accessible. Because there is no point in making medicine which, even if it is effective, people cannot afford. And I think that is the situation now with cell therapies. They are expensive, very expensive, and complex, and also generally inaccessible to many patients. So we think that the way to make it affordable is actually, it's got to be allogenic. In other words, we use donor blood to make the cells, right? So use donor, not the patient's own blood. Many of the companies actually now use autologous or donor's blood. Patient's own blood, or they modify it, they engineer it so that it becomes allogenic. As you know, we announced yesterday we bought a licensed cord blood bank at a very affordable price, less than $500,000. Right, and this is something very exciting for us because we intend to use this cord blood to make allogenic T and NK cells. Now, cord blood has its own unique immune properties. So, cord blood-derived immune cells will complement our existing repertoire of immune cells. Right, that we now actually make good gamma delta T cells, right, allogenic from donor blood, we make it from adult blood, and also induced pluripotent stem cells, right? So we are good at that now from adult. So this cord blood acquisition will enable us to make it from cord blood babies, right? So pristine condition. So we hope to be the master of allogenic donor T cells and NK, right? Making them from three main sources, which is blood, adult blood, iPSC, and soon from baby, the cord blood. So the other way to make it affordable and accessible to patients is, second thing is, so first is we're going to make it from donor. And secondly, we hope to use Asia. We're going to use Asia as a low-cost launching pad, economic base, to make cell therapies accessible to more people. Why? Because we operate in Southeast Asia. Our cost infrastructure is significantly lower. As a good example, we can even own our own real estate. We own all our properties building the lab, because once you build the lab, you cannot change location. So we are a strange animal. We actually get to own our own facility, right? We pay for it and we own it, and that's because we operate in Southeast Asia, and our lab is CGMP, PIC standard. I mean, remuneration in Asia is significantly below the West also. As you know, in India, you can really appreciate the huge treatment price differences. India makes CAR-T, open heart surgery at very low cost. So similarly, I think Southeast Asia is where we're going to make it accessible, make our cell therapy accessible and affordable. In fact, Dr. Tan and I, we are flying off to India tomorrow morning, first thing tomorrow morning. Right. So I think we're going to show you two videos. You look at the circled yellow in yellow. That large ones are the cancer cells. You might see it, growing. One become two, two become four, and the small ones you see there are the T cells that we make, right? The special gamma delta T cells, delta 2, by the way. And this is in vitro lab photo. There are two videos. One is the second video has a lot more cancer cell and with higher dose. Right? So you can see the end effect is to see the clearing of the field. Right, the circle field becomes clear. Right. So Ryan, would you play the video, please?
Okay, right. Thank you, Peter. Now, let me share about the company as well as to run through the technology that we are using. We are headquartered and incorporated in Singapore back in March 2018, and we are spun out of A*STAR, which stands for Singapore's Agency for Science, Technology and Research. And that is a government-funded agency that does research into deep tech, fintech, AI, and biotech. We are currently preclinical. However, we have already started our first in human phase 1 clinical trial that I will touch on a little bit more. Now, what set us apart is that we use healthy donor blood and not from sick patient. If you were to look at the slide on the left-hand side, everybody will be very familiar with the autologous approach of typical CAR T cells, where you have to wait for the patient to come in, harvest their cells, and you can only give it back to the patient. If you give it to another patient, it will actually cause graft versus host disease that would be very serious and may be even potentially fatal. And the reason for this challenge is because they use something called an alpha beta T cell. All of us have two types of T cells, with the alpha beta T cells constituting about 95%, and the gamma delta T cells being at 1 to 5%. The alpha beta T cells are abundant in their numbers, and that's why it's easy to harvest and make use of them. But the challenge is that it can only be used in an autologous setting. For gamma delta T cells, however, which is the cell type that we focus on, it could be obtained from a healthy donor, and that means that we have a much better starting material quality. It could be massively expanded outside of the body to very high purity and in numbers, and it could be infused back into many different patients without the need for matching. And that enable us to treat many different patients, potentially making it effector cells of choice for allogeneic therapy. The second advantage of using gamma delta T cells is the fact that the gamma delta T can recognize 8 different NKG2D ligands, which are expressed on a wide variety of cancers. And you can see here this is a non-exhaustive list. The list is longer on the cancers that actually expresses NKG2D ligands. So over here, you can see the typical leukemias, the lymphoma, the multiple myeloma, which all of us are more familiar with, especially in the CAR T space. However, excitingly, the gamma delta T cells can recognize solid tumors, as you can see here as well. So in fact, most of the cancers, the large proportion of cancers are actually solid tumors as compared to leukemias. And therefore, this means that it blows the market wide open, as you're able to treat many different indications with one cell type, and it can be used in many different patients without matching. So this is a feature, a newspaper feature of our first in-human phase 1 clinical trial with a leading cancer hospital in Singapore. Our clinical collaborator is National University Hospital. And it is exciting because our first in-human trial has been approved for 6 different indications, of which two of it is blood disorders and 4 solid tumors, meaning that both the regulator in Singapore as well as our clinical collaborator have strong and firm belief in the fact that the cells are able to recognize multiple indications. So our first in-human trial has already begun. It is being carried out in 2 parts. The first part would be collecting adequate blood donors, which we have already done so. And we would be moving as soon as possible into recruiting our patient to start the clinical trial. So let's jump in and take a look at the pipeline. We have the gamma delta T cells that are allogeneic and unmodified. We have the CAR gamma delta T cells that we have spoke about just now. And for your information, the CAR gamma delta T cell, the CAR that we're working with, is also an NKG2D CAR. And we have a next generation pipeline that is in development, that is our iPSC-derived gamma delta NKT cell, and that's in GMP development. We list the gamma delta T and the CAR gamma delta T cell as GMP ready. That is because we have the clinical protocol all worked out, the GMP production capabilities all done up, and we are able to produce clinical grade, GMP grade cells in-house that I will touch on a little bit more. Our iPSC gamma delta NKT that is in GMP development, but process development is ongoing, but proof of concept, including publication and patent filing, has been done. To have a brief rundown of what is iPSC, iPSC stands for Induced Pluripotent Stem Cells. And induced pluripotent stem cells is a type of cell that has been engineered from an adult. So initially, people work with human embryonic stem cell that is being derived from the blastocyte, but that contains ethical concerns and challenges. So an induced pluripotent stem cell is here, where you take a cell from a consent-giving adult. It could be reprogrammed with transcription factors, and you can put it back into the iPSC, and you can turn it back into the iPSCs, which could be differentiated into different types of cells. For us, because we have iPSCs, they are able to renew limitlessly, and for the next generation platform, we will not need a donor anymore. We are able to produce a 2-in-1 hybrid cell, combining the gamma delta and the NK cells together. And these cells will give you an enlarged repertoire of receptors that enable you to recognize different types of cells. For our clinical pipeline, as I mentioned, the CAR gamma delta T, we are moving into phase 1 already, with the donor recruitment in progress, but we have adequate donors, and we will be doing patient enrollment as soon as possible. iPSC gamma delta NKT, we are targeting preclinical studies after 2024. And for our unmodified gamma delta T, we are looking at US FDA IND application and trial application in Malaysia. We have also been working with MD Anderson Cancer Center for preclinical studies, and we have already filed our DMF with the US FDA. Quick touch on our streamlined manufacturing. Below is a typical process of a conventional CAR grafted alpha beta T cells. You can see that there are challenges, including limited numbers, poor starting cell quality, and the potential of cancer cell contamination because it's taken from a cancer patient. For us, it's much simpler. We would have the cells from the donors ready. We will be able to produce a large amount of cells with good cell quality, and there's no contamination of cancer cells. And so because of this, we can produce a more standardized cell, and these cells would not require a long waiting time. It could be made readily available, and so it becomes an off-the-shelf allogeneic therapy. Where we stand in terms of patent filing, our first IP that covers the gamma delta and the CAR gamma delta T has been granted in US, China, and Malaysia. And the second patent, which is on iPSC-derived gamma delta NKT, that is covered, we have it granted in Japan, China, and Malaysia. And finally, we own and operate our own PICS GMP manufacturing facility that has been audited by health authorities in Malaysia. We are using it to support the phase 1 clinical trial in Singapore, and it is well-staffed, well-equipped. And so we are a biotech that is very unique because we possess capability to do in-house manufacturing, in-house trial application, and we own all our assets, which you can see here, which is here we are headquartered in Singapore, and we have our PICS GMP lab just across the border in Johor, Malaysia, and we have also a lab in the capital city of Malaysia, Kuala Lumpur. And so with that, I will pass it back to Mr. Peter Choo.
Well, in this biotech winter, our share price is about 50% below our IPO price. We listed in April last year. Not a good time, because at that time the US banks were having trouble, interest rate high. So our IPO price was $4. Now we're about $2. Our market cap is about $20 million, 20. But there are a few occasions where the share price stayed above IPO price, and it's hovered around $2 now. But I want to assure my shareholders that I will be working very hard. I'm fully aware of the situation, and that's why I'm coming to New York. Right, so I'll be working hard on many ideas, right, to not to disappoint my shareholders. Right, so I wanted to show you our financial performance last year. Right, if you basically all this information is in our SEC reporting. And I wanted to say that if you exclude the IPO expenses, the underwriting, the legal auditing, our loss last year for 2023, right, our first year of since listing, we lost $2 million, right? Which I think compared to many biotechs, I think it's it's respectable. I would like to think that way, right? We lost $2 million. And then that's really because of our low-cost infrastructure in Asia, right? And we IPO'd raising only about $8 million net. We still have a bit of cash, still a good size of cash, as again due to the low-cost infrastructure in Asia. Our shares are quite tightly held. There's 11.5 million shares only. So if you times 2, we're about $22 million market cap. Right? So I wanted to show you this also. The market comparable. We are also in the space of iPSC, which is now dominated by companies like Fate, Century. They are respectable, good companies, and with a good market cap, right? So I just want to highlight this to reflect our market cap of 20 million or 22 million, which I would feel that we are really underappreciated. And that's because we operate in Asia. So now this is my last slide. We seek strategic partners, especially in US, right? So that we can be heard in the US. So we are going, our market, we are going for Southeast Asia, population 700 million, China 1.3, India 1.3. So our direction will be Southeast Asia, China, and India, right? So with that, I would end my presentation. And if you have got any question, either you email us or you can ask now. Thank you very much. Thank you.
Wonderful. Thank you both so much for your presentation. We do have some questions for you. Reed wants to know if you've seen any complications or side effects, even with the same blood type after your therapeutics is added.
Right. So we have, while we have not started the clinical trial, we have actually done some, we have tested preliminarily on patients that has no options. And even if the patients and the donors are of different blood types, we do not see any complication at all. And this sets the therapy apart very much because of its well-tolerated and safety profile, but we are able to see some promising efficacy. So meaning that we are able to split away the side effect versus the efficacy, which is typically not seen in cell therapy.
And with that, Lincoln wants you to explain what type of blood you're using primarily. Is it cord blood, donor blood? Explain what you use each for.
Right. So we Our platform works with adult peripheral blood. So the process is very simple. It's like you're going to any blood bank to donate the blood. So we screen the healthy donors to ensure that they are free of infectious disease before we take their blood for further processing. So that is our starting material that we are using for our gamma delta T, our current gamma delta T, and even to reprogram into iPSCs. For the cord blood, we have acquired a company, and we will be developing a cord blood derived cell therapy, particularly gamma delta and NKT from cord blood in the near future.
All right. And Cohen asks if you can talk about the approval process for your therapies, both domestically and abroad.
Right. So in Singapore and abroad, I think the approval process is pretty similar. You could hasten your process by talking to the regulators early, which is what we did. And after that, you put in the filing as standard, your CMC filing, your investigator brochure, your clinical protocol. And once they review it and approve it, you're ready to go. So in Singapore and in other markets like US, you have accelerated approval or conditional marketing that enable you to quickly commercialize if your results are good post phase 2.
I would like to add that the Southeast Asia, many of the health departments are very keen to go into this new science of cell therapy, right? Gene editing is fine, but I think it's a bit more new. But I think a lot of the governments in Southeast Asia, Thailand especially, Thailand is number one now in medical tourism, Malaysia, Singapore, Indonesia. Southeast Asia is jumping into this new science, and the governments and the ministry have all announced ideas and plans to develop cell therapy.
Thank you for that. Vince says, when you say you're funded, how is that structured? Is it in equity, debt, a grant? How does it work?
We have no debt. It's funded by private angel investors initially. As far as I know, I don't think we have got institutional money, because every one of us know is biotech winter. So we fund on private angel money, and our IPO money, although it's meager, but it is a lot of money to us, right? That $8 million that we raised net from the IPO is a lot of money. We still have got half. So, and that's really helped by the low cost here. So we don't have any serious institutional money, right? As far as I can look at my list. So it's private money, no debt.
Perfect. And Emery says he likes how you do everything in-house. So do you have any sales currently? And what is your timeline to sales?
We don't have a sales department because the products are not approved. They're all still in research. But every now and then we have got medical people, hospitals emailing us for help, right? So under such compassionate, no option, unmet need situation, after discussing with the doctors, we do, discuss how to handle their case. Right? So we do have emails from even the US. But as I said, our trial will start soon, right? Because we have now finished with the donor blood. So, because our trial, we are quite unusual because we need to do the first part, which is getting the donor. We're going to find good donors, right? And then we do the second part, which is the recipient. So we are going to start our trial soon. And with that, I think we should be able to talk to, more hospitals will approach us, put it this way, yeah.
Perfect. And in general terms, talk about how you are different from other listed biotechs.
Right. So I think I think what sets us apart, a few levels. One is the bulk of cell therapy focuses on autologous therapy, while we, throughout our pipeline, we focus on the same theme of it being allogenic, and that means that we are able to produce the cells at a lower cost and a shorter waiting time for all our patients. And we hope that this would help the patients access the drug faster, as well as for more people to access the drug. And I think what sets us apart really is the fact that we are operating in Asia. That enables us to tap into a huge population within Asia, but operating at an extremely cost-efficient manner, which is as per our annual report. And the fact that because we are able to do that, we can control everything in-house, with in-house manufacturing, in-house clinical filing application, right? And because ours is allogenic, at some point in time, it will be distribution or supporting multiple site trial within a centralized manufacturing facility. So I think these are the few things that really set us apart and enable us to tap strongly and deeply into the Asia market.
Wonderful. Well, that's a great way to end the presentation. We are out of time, but really great job, both of you. And thank you so much for this important work you're doing. And we certainly hope you come back on the conference to give us some updates.
Thank you very much, everyone. Thank you.
Thank you for having us. Thank you.
All right. Have a great rest of your evening. Everyone stay with us. We'll be right back.